Hi all. Today we are going to discuss about the topic variations in genetic code in Mycoplasma and Tetrahymena. We have already studied about what is genetic code and what are the characteristics of it. Its characteristics involves universality, commaless, non-ambiguity, non-overlapping, stop codon, start codon, etc. But there are some exceptions in this genetic code. In this topic, we are discussing about the exception in genetic code. We studied that genetic code is universal in nature. But in spite of universality of genetic code, there are some exceptions. The exceptions are mainly concerned with the codon for initiation and termination. Now, we are discussing how the genetic code of Mycoplasma and Tetrahymena vary from others. Mycoplasma is a genus of bacteria that lack cell wall. They can be parasitic or saprotrophic. You know that Mycoplasma is a bacteria but they does not have any cell wall. In case of Tetrahymena, it is a genus of free-living ciliated that can also switch from commensalistic to pathogenic mode of survival. First, we can discuss about Mycoplasma's genetic variations or variation in its genetic code. The first point is that Mycoplasma have a reduced genome size. We have already said that Mycoplasma is a genus of bacteria, but comparing to other bacteria, Mycoplasma species have small genome. It is due to the loss of many genes that are essential for bacteria. For example, Mycoplasma genitalia. It has only 482 protein coding genes. It is very less when compared to other bacteria. Second and important variation in genetic code is that it is non-universal in nature. We studied that genetic code is largely universal, but in some Mycoplasma species, they shows variation. In Mycoplasma capricolum, UGA, which is usually codes for termination, not used for termination in Mycoplasma capricolum. Instead of that, UGA codes for the protein tryptophan, and it is more preferred than the usual UGG code. UGA, which read as UAA and UGG, that is tryptophan. CCA, which read UGG. Then Mycoplasma species have high mutation rate. We studied that there are so many DNA repair methods in our body and other eukaryotic and prokaryotic body. But in the case of Mycoplasma, some species have lack of many DNA repair method. Due to this, they shows high rate of mutation. This mutation will lead to genetic variation and evolution of species. Mycoplasma species have another ability to differential gene expression. They can modulate their expression according to the environmental cues. Environmental cues such as nutrient availability or temperature, etc. So they produce different protein in different environment because they have the ability to modulate their gene expression in response to their environmental cues. Then horizontal gene transfer. Mycoplasma shows a horizontal pattern of gene transfer. They are known to acquire gene through horizontal gene transfer method, which means that can acquire gene from other bacteria or even from the host. On the first slide, we have discussed that Mycoplasma does not have cell wall. This is due to the loss of genes which codes for cell wall biosynthesis pathway. Other bacteria have cell wall, but in case of Mycoplasma, They lack cell wall due to the loss of gene encoding for cell wall biosynthesis pathway. Then they have initiation sequence. Mycoplasma species contain insertion sequence which are mobile genetic material. This mobile genetic material can move within the genome. This result in the genetic variation and genome evolution. Hope you understand. Insertion sequence is a mobile genetic material which can move within the genome. So, the movement of this sequence will lead to genetic variation and genome evolution. Now we can study about tetrahymena. We said that mycoplasma have a reduced genome size. But in tetrahymena, there is an opposite condition that they have a large genome size. Tetrahymena species have large genome when compared to other single-cell eukaryote. Tetrahymena is a ciliated single-cell eukaryote. But apart from other single-cell eukaryote, tetrahymena shows large genome size. Genus of Tetrahymena thermophila contain over 24,000 protein coding genes, which is very higher when compared to others. Then Tetrahymena shows alternative splicing, which means they can produce multiple protein from a single gene. This method of splicing is known as alternative splicing. They are present in Tetrahymena. Then presence of transposable elements. Tetrahymena have high frequency of transposable elements such as signs, lines, etc. This contributes genetic diversity and genome evolution. Then endosymbiotic gene transfer. They undergoes endosymbiotic gene transfer in which gene from endosymbiotic organisms such as chloroplast genes, mitochondrial genes, that is chloroplastal genome or mitochondrial DNA are transferred. In case of genetic code, Tetrahymena use only a single codon UGA for the termination of protein synthesis. In Tetrahymena thermophila, UAA and UAG is not used for termination. UAA and UAG codes for the amino acid glutamine. UUG, which read usual codon on CAA and CAG for glutamine, where UUA, which read as UAA and UAG. CUA, which read only UAG. Point mutation that alter genetic codon. This is the next topic we have to discuss, that is point mutation that alter genetic code. From the name itself, it is clear that point mutation is a single change in the nucleotide of a genetic code, which causes alteration of genetic information of an organism. So, point mutation are the changes that occur in a single nucleotide within the genetic code which lead to alteration of genetic information of an organism. Point mutations effect depend upon where they occur and what the type of mutation is it. So, its effect varies according to its location and its type. Mainly, point mutation are of two types, substitution and frame shift mutation. Substituted mutation, from the name itself, it is clear that where a nucleotide is changed, it is replaced by another set of nucleotide. In substitute mutation, there will be change of a nucleotide or a few nucleotide from one type to another. One is substituted by other. In this substitute mutation type, there are three types of mutation: silent mutation, missense mutation, and nonsense mutation. So, first we are discussing about silent mutation. Silent mutation does not affect the amino acid sequence of protein, as the change in the nucleotide does not change corresponding code. From the name itself, it is clear that it is a silent mutation. That means they does not have effect on the amino acid sequence of protein because the change in the nucleotide does not change the corresponding codon. So they does not have any effect on the proteins. Next is missense mutation. Missense mutation is a type of point mutation that occur when a single nucleotide change within a gene lead to the different amino acids incorporated into the protein sequence. We study that point mutation is a change in a single nucleotide. So, in missense mutation, a single nucleotide which lead to the different amino acid protein synthesis. It can either benign or harmful. Some mutations does not have effect on protein structure and function, but some missense mutation will lead to diseases like cancers, neurodegenerative disorder and metabolic disorders. But some mutations are beneficial because that provide resistance to certain diseases. This missense mutation can be identified through the process of DNA sequencing. Missense mutation may be spontaneous or inherited. It also affects in splicing. It causes genetic diversity and variation. Sickle cell anemia is a perfect example for missense mutation. In sickle cell anemia, it is caused by a single point mutation in beta hemoglobin gene that convert GAG to GTG, which encode valine rather than glutamic acid. We can clearly understand from this figure. In normal hemoglobin chain, GAG, while mutation occur, this A is replaced by T, which leads to, instead of glutamine, they synthesize the protein valine. This leads to the sickle cell shape of the RBC and reduce the oxygen carrying capacity of the hemoglobin. This is the cause of sickle cell anemia, that is missense mutation. Next one is nonsense mutation. Nonsense mutation is a type of point mutation that occur when a single nucleotide change within the gene which create a premature stop codon. A single change which leads to synthesis of a premature stop codon. This results in the termination of translation and production of truncated protein. This nonsense mutation leads to the creation of premature stop codon, which stop the termination, which cause termination of translation and leads to the production of truncated protein. This mutation have severe consequence on protein function because the truncated protein have a different function and which are unstable. This cause genetic diseases. Example, cystic fibrosis, hemophilia, muscular dystrophy, even it can lead to cancer cell and contribute tumorigenesis. This nonsense mutation also can be detected using DNA sequencing technique. It may be spontaneous or inherited. It reduces genetic radiation and diversity. Even nonsense mutation occur in gene that encode CFTR protein. It can lead to cystic fibrosis. Nonsense mutation in the CFTR protein is the reason for the disease cystic fibrosis. We can compare missense mutation, nonsense mutation and silent mutation. Missense mutation, a change in a nucleotide, a single nucleotide leads to the production of a different protein from the normal one. Wild type is AAT, which codes for the amino acid leucine. Due to missense mutation in AAT, T is replaced by adenine A, then which result in the production of an amino acid phenylalanine. Phenylalanine instead of leucine. A single change in the nucleotide leads to the different protein synthesis. Here T is replaced by A, which result in the production of phenylalanine instead of leucine. Nonsense mutation, we studied that. Nonsense mutation, change in a single nucleotide leads to the synthesis of a premature stop codon and which result in the truncated protein synthesis. So in this case, AAT in place of second position A, it is replaced by T, which result in the codon ATT. In RNA, it's transcribed as UAA, which is act as a stop codon. Then silent mutation, which does not affect on the protein sequence. Here AAT is the wild type. In after silent mutation, it becomes AAC. But when its RNA is transcribed, it is codes for the protein leucine, which is similar to the wild type. Then second type of point mutation is frame shift mutation. Frame shift mutation occur when one or more nucleotide are inserted or deleted from a DNA sequence that is not a multiple of 3. This will alter the codon on reading frame, downstream of mutation leading to the translation of completely different amino acid. Here, one or more nucleotides are inserted or sometimes it is deleted, which is not a multiple of 3. So, this will lead to the translation of completely different amino acid sequence. It have severe consequences on protein function because they introduce premature stop codon or it shift the reading frame. The shifting of reading frame leads to the production of completely different protein. It can also occur in different non-coding region of genome, which effective gene regulation, chromatin structure or gene stability. Frameshift mutation also causes gene regulations, chromatin structure and gene stability. It is also affected by frameshift mutation. It also causes genetic diseases. Example, Tay-Sachs disease, Huntington's disease, etc. We can discuss how Tay-Sachs disease occurs. It is due to the mutation of hexa gene which leads to the Tay-Sachs disease. There are more than 80 such mutations have been identified. But one of the common cause of Tay-Sachs disease is that single nucleotide deletion which leads frameshift mutation. We have studied that frameshift mutation is the addition or deletion of a nucleotide or a few nucleotides which are not multiple of 3. So, in case of this disease, the 1510th nucleotide of cysteine is removed and which leads to the frameshift mutation and causes Tay-Sachs diseases. From this figure, we can understand frameshift mutation. On the first row, it is normal mRNA polypeptide chain. Then second one shows how insertion, that means insertion, addition of a nucleotide, how it affect the normal one. The third one shows how deletion of a nucleotide affect the normal one. Normal one is given above. By the addition of the nucleotide U, the frame shift get changed and the codon is as follows: AUG, GGC, GCC, AAA. Instead of AGU, here U is introduced. Then it becomes UAG and U. Normal polypeptide chain U got changed into next frame as UUA. So instead of serine, here it codes for a stop codon or a termination codon. Okay, then deletion. How deletion of a single nucleotide affect the frame shift mutation, cause the frame shift mutation. Here normal sequence is AUG, GGG, GCC, AAA. When G get deleted from the sequence becomes AUG, GGC, then G is removed. Then it becomes CCA, which codes for proline. Instead of alanine, it codes for the protein proline. That means it codes completely different protein. This is the examples for frame shift mutation. Hope you understand. Thank you.